Hello everyone. Today we will study about insulin regulation. So insulin can regulate metabolism and gene expression. Metabolism means anabolic and catabolic activity, and gene expression means it can transcribe gene. Okay, and it can transcribe gene and produce a kind of protein. Okay, so what happening? Insulin it it bind to the actually in human body in there is many kind of signaling sig- a many kind of receptor is present that can bind to the outsider molecule, and on the basis of their binding, when it bind, then there the inside cellular enzyme activity will start. Okay, so insulin is also such type of things. Okay, in insulin, what happened? See, this is the plasma membrane. Okay, this is plasma membrane. So this is the receptor, and these receptors are very specific. This is transmembrane, and inside this is the another subunit of this receptor. So it having two subunit. One is the extracellular side, cellular side, and another is the cytosolic. Okay, two sides are there. So what happened? I just take a different color. So when ligand bind to here, suppose this is the ligand. When ligand bind to this receptor, inside inside the cell, some changes inside this receptor, some changes will happen. It will phosphorylate either by the GTP, by ATP, or by itself. Autophosphorylation will happen. When this phosphorylate, then there the active site, it will activate the another kind of enzyme. Okay, so that we have to study. So in human body, the signal is transferred to the cell and to the nucleus by the many ways, by the carrier protein, by the channel protein, by the G protein, and the receptor enzyme. So here we have to study about the receptor enzyme, and the one example I have taken is the insulin regulation. So receptor enzyme is what? A protein that a protein have bind ligand on the extracellular surface, and the cytosolic side and the enzyme active cytosolic side of the domain, both are connected by the transmembrane, as you can see here. So that is the receptor enzyme. Okay, now see the insulin receptor. If you check the insulin receptor, what happening? So this is the insulin receptor. It having two alpha chain and two beta subunit. Okay, so in alpha subunit, insulin will bind to it, and when the insulin will bind, then in the beta subunit, tyrosine will phosphorylated with the help of tyrosine kinase. Okay, and how many they can? So minimum three tyrosine get phosphorylate, though then only the further another kind of protein kinase will get active. Okay, so this is the one condition. Okay, this is the insulin receptor. Now what happened? See this picture I have taken from Lehi Ninja textbook, so you can check there. So what happened? As it is see here, this is the alpha subunit, this is the beta subunit. When insulin want to eat at the alpha side, beta subunit get phosphorylate, autophosphorylation, and when this phosphorylate, it activate another kind of enzyme, another kind of receptor, another kind of enzyme. Actually, the name of this is insulin IRS1, insulin receptor substrate. Okay, and when it phosphorylates, it phosphorylate many enzymes, and ultimate this information will reach to the nucleus, and it will when it will reach to nucleus, it will transcribe a kind of protein. Okay, so this is the process of insulin, how insulin regulates gene expression. Okay, so we will look one by one what are these things. What happening actually? See, this is the plasma membrane. This is the plasma membrane, and here is the insulin receptor. So insulin bind to it, and beta subunit will get phosphorylate. Beta subunit get phosphorylate. When this phosphorylate, the one enzyme, the name is IRS1. What I told, insulin receptor substrate. So it will phosphorylate. It will phosphorylate. IRS, it phosphorylate. Okay, it phosphorylate. And when it phosphorylate another kind of enzyme. The name of enzyme is GRB2. GRB2. Okay. So what happened? GRB2 bind the bind IRS1 with the SH2 domain. Suppose this is the SH2 domain. So this is what? This is SH2 domain. Let's check the color. This is what? H2 domain. What happening? SH2 domain in many enzymes are present in the cytoplasm that having SH2 domain. Okay, suppose this is a cell. See, suppose this is the enzyme. Any enzyme is this, and suppose this is the GRB only. GRB2. What happened? This is the SH2 domain of GRB. Here is the also SH2 domain of GRB. This is also SH2 domain of GRB. And check in the different color. This is the SH3 domain of GRB. This is the different binding site for the different protein. Okay, enzyme have different binding site for different kind of protein. So what I told, this is SH3 domain. SH3, and this is what? SH2. Okay, so these are the things are present. So when IRS1 phosphorylate, it can activate many SH2 domain binding protein. Okay, so one of the example is GRB2. Now GRB2 SH2 domain bind to phosphorus group of the IRS and it activated. It activate, and what happened? It having another binding site also. That binding site is called SH3 domain. SH2 means what? Sarc homology 2. Sarc homology 3. That is SH3 domain. SH3 domain. Okay. Now this SH3 domain bind with another protein. The name of the protein is, suppose make such type of thing. Another kind of protein. Name of that protein is SOS. SOS. So it bind with this. So this is the this receptor on the SOS protein. It bind with the SH3 domain here. Okay, make a little bigger like this. It bind with the SH3 domain. Okay. So now what happened? This source protein is activated. And what happened once this source protein is activated, it will activate another kind of protein. The protein name is RAS protein, and it is a G protein. It's a RAS protein. And when this active, the G protein means it will take GTP and get activated. Okay, convert to GDP. Okay. Once RAS protein is active, now it will activate. Now this will activate MAP kinase. MAP kinase. Kinase, all the range of protein it will activate. And what is this MAP? MAP means, here I am writing, mutazine. It's a mitosine, mitosine activated, activated, mitosine activated protein kinase. Protein kinase. What is the meaning of this big line? Mitosine activated protein kinase. Mitosine is something that has capacity to start mitosis or cell division, or cell division. It has capacity to start. Okay, so such a molecule, that's the outsider molecule, the outside of the cell, that bind to the receptor and it activate the transcription. That is mitosine. Okay, mitosine are the signal that are that act from the outside of the cell to induce mitosis or cell division. Okay, so now here what happened? RAS protein activate, and RAS protein will activate MAC kinase. Okay, so the next protein is RAF. Here RAF, RAF1, then is the MAC, then is ERK, K, and then the final will be E, ELK, ELK1. Okay, so this RAF will first activate by the RAS, and that comes under protein kinase. MEC comes under mitosine activated protein kinase. ERK comes under kinase. Okay, these two continuous phosphorylate by this sequence only, and this ERK will reach to the nucleus. It can get inside in the nucleus, as you can see in the picture, as you can see here. ERK get inside here and it transcribe ELK1. Okay, it activates another protein, that is ELK1, and that ELK will bind to DNA and transcribe, and it can transcribe 100 regulatory gene. Okay, so this is all about the gene regulation, gene expression by insulin. Okay, how first the IRS1, then the GRB2, then the SOS, then the RAS, then the RAF, then the MEK, then the ERK, then ELK. This is the sequence. Okay, if you will, if you're having any confusion here, you can check in. Glycogen regulation we will see in the next video. Okay, I think up to there how insulin express the gene activity you understood, right? And next we will see glycogen regulation. Thank you.